Hey there. Today I want to talk about the basics of cellular respiration, the three major steps of cellular respiration, the products that are being made, and why the cell is going through all this trouble in the first place. And to do that, I'm going to use a couple figures from this textbook here called A Visual Analogy Guide to Human Anatomy and Physiology by Paul Krieger. And I really enjoy this textbook. As the name implies, it utilizes a lot of analogies to learn the different parts and processes of anatomy and physiology, and I think it's a really good way to learn human anatomy and physiology. And let's start by writing out the equation for cellular respiration. What's the equation for cellular respiration? Well, we start off with a molecule that is C6H12O6. What molecule is that? That's glucose, and we're going to combine glucose with 6 oxygen molecules, and then we're going to get some products. What products are we going to get from this? Well, we're going to get 6 carbon dioxide molecules. We're going to get 6 water molecules, and most importantly, we're going to get about 38 ATP molecules, 38 molecules of adenosine triphosphate. And this is the whole reason that we're performing cellular respiration, is that the cell needs to produce ATP. Why is ATP so important for the cell? Well, all living things, all cells require chemical energy to power their activities. And the form that chemical energy needs to be in for the cells to be able to utilize is ATP. So we're converting energy that we obtain from nutrients like glucose, and we're converting that to a form that the cell can use, ATP. Now there's also one other product here that I haven't mentioned. What's that last product? We're also putting off heat in this process, and it's this heat that is heating the body. It's the reason that we can maintain constant and elevated body temperatures in humans of about 37 degrees Celsius. All right, let's give this thing a go. Here's our glucose molecule right here, and we could draw a blood vessel. We obtain glucose from the diet. It gets absorbed from the digestive tract and enters the blood, and then it diffuses out of capillary beds into the extracellular fluid, into the interstitial fluid. Now what we have to do, though, is we have to get that glucose molecule from the interstitial fluid, and we have to get that inside of the cell. How is that going to happen? Let's draw ourselves a little section of the plasma membrane. There we have the cell membrane. And what's the cell membrane made up of? It's made up of phospholipids. It's a phospholipid bilayer. It has 2 layers of phospholipids, which consist of glycerol heads and fatty acid tails. And this glucose molecule has to move through this plasma membrane to get into the cell. And again, how is it going to do this? Well, let's throw out some options here. One of the options for glucose would be simple diffusion. And simple diffusion simply means that glucose molecule is going to diffuse through the plasma membrane on its own to get inside the cell. Now, is that something that glucose can do? That's not. That's not an option for glucose. And why not? Glucose is simply too big. Okay, glucose is a large molecule, and also I doubt that it's lipophilic. It's likely lipophobic. And so both of those reasons preclude it from going through the plasma membrane. So what do we have to have in the plasma membrane to get that across? we have to have a carrier protein. So I'll try to draw a carrier protein here. And there's our glucose molecule. And what's going to happen is this glucose molecule is going to bind to the binding site on this carrier protein. That's going to cause the carrier protein to change shape. And that's going to allow the carrier protein to release this glucose molecule inside of the cell. And this is occurring by diffusion. Glucose is moving from an area of higher concentration outside the cell to a lower concentration inside the cell. And what do we call that type of diffusion? That's called carrier-mediated diffusion. Clean this up a little bit here. Okay. So now we have glucose inside of the cell. And this is where we begin the first step of cellular respiration, which is glycolysis. Now, what's happening with glycolysis? With glycolysis, we're taking this glucose molecule, which has 6 carbons in it. That's what each of these little spheres is representing there. And we're breaking it down into 2 pyruvate molecules, each of which have 3 carbons. So we're taking a 6-carbon glucose and we're undergoing a catabolic reaction and we're breaking it down into 2 pyruvate molecules. And those pyruvate molecules are going to move on to the next step of cellular respiration. Now, what else are we producing in this process? We're making some ATP. Are we making a lot of ATP? No. We're only making 2 ATP here. There's about 38 ATP adenosine triphosphates made total from 1 glucose molecule, and we're only making 2 here. What else is glycolysis giving off? It's giving off some electrons. And what's going to happen with these electrons is they're picked up by these molecules called NAD+. And after they pick up those molecules, they become NADH. And NADH is what we call an electron carrier. And it's going to shuttle these electrons to the last step of cellular respiration, where they're going to be involved in producing mass amounts of adenosine triphosphate. A couple other things I want to point out here or address. Where in the cell is glycolysis occurring? Is it occurring within an organelle? It's not. Glycolysis is occurring in the fluid of the cell, in the cytosol of the cell. Something else, cellular respiration requires oxygen, but is glycolysis using any oxygen? It's not. There's no oxygen involved, and so that makes glycolysis an anaerobic process. And it's the fact that this is an anaerobic process that prevents us from producing large amounts of ATP. To produce large amounts of ATP, as we'll see, that requires oxygen. Now what happens is those pyruvate molecules move into this structure right here. And what is that structure called? What is that organelle? That's the mitochondria. So the pyruvate molecules move into the mitochondria. One of those carbons is clipped off and they become a molecule called acetyl-CoA. And it's the acetyl-CoA that's going to enter the second step of cellular respiration called the citric acid cycle or the Krebs cycle. So acetyl-CoA enters into the Krebs cycle, and it goes through this complex series of metabolic events that we're not necessarily going to talk about, but what I do want to look at are the products. What are the products that are being made by the citric acid cycle? Well, again, we're making some ATP down here. Are we making a lot of ATP? No. Again, we're only making about 2 ATP here. The main product from the citric acid cycle are going to be these NADH molecules, these electron carriers. We're going to produce about 6 NADH molecules, and we're also going to produce another couple electron carriers called FADH2. So we're making NADH and also FADH2, and both of those are electron carriers. And again, what's going to happen with those electron carriers is they're going to deliver those electrons to the final step of cellular respiration, where all of that ATP is going to be generated. What else are we producing with the citric acid cycle? We're producing carbon dioxide. And in fact, just converting pyruvate to acetyl-CoA produces carbon dioxide. And if we look back at our equation for cellular respiration, sure enough, there it is. One of the products is carbon dioxide. Now, carbon dioxide has to be removed from the body at the same rate that it's being formed. If too much CO2 accumulates in the body, then the body becomes acidic, and that leads to major problems. So we have to get rid of this CO2. And that's what this is showing. CO2 moves out of the mitochondria, and what's going to happen is it's going to diffuse out of the cell. Now, how does carbon dioxide diffuse out of the cell? Well, that occurs by simple diffusion. Carbon dioxide is a small molecule. It's a small gaseous molecule, and that means that it can diffuse by itself through the plasma membrane. It doesn't need any help to do that. And maybe a better way to do that would be to draw a whole bunch of these CO2 molecules here. Because with diffusion, molecules are moving from an area of higher concentration to an area of lower concentration. And so carbon dioxide simply diffuses out of the plasma membrane. Then it moves into the blood, and where does it go next? It's going to go to the lungs, and it's going to diffuse out of the respiratory capillaries, out of the pulmonary capillaries, into the lungs, and we exhale it. That's how we get rid of that carbon dioxide from the body. And now we're going to move on to the third and final step of cellular respiration, which is the electron transport system. And what's going to happen with the electron transport system is those electron carriers that we produced in previous steps are going to make their way to the membrane of the mitochondria. Now, the mitochondria is an organelle, and all organelles are surrounded by a plasma membrane. And in fact, there's going to be an outer mitochondrial membrane, an inner mitochondrial membrane, and then a space in between them called the intermembrane space. And this is where the electron transport system is going to occur, but this will be more clear on the next figure that I show. And what's going to happen is these electron carriers are going to drop off these electrons, and the electrons are going to be used to produce large amounts of ATP. The electron transport system produces large amounts of ATP. And about how many are going to be produced? We're going to have about 32 of those molecules made during this process. So by far the vast majority of ATP is produced in the electron transport system. So what haven't we accounted for yet in our cellular respiration equation? We haven't accounted for oxygen. So now we're going to have oxygen that's involved. And what's going to happen is oxygen is going to enter the cell the same way that carbon dioxide exits the cell, by simple diffusion. Oxygen is a small molecule, and it can pass straight through the plasma membrane. And it's going to do that by moving from an area of higher concentration to an area of lower concentration. And again, that's called simple diffusion. And then what you can see is that oxygen moves down into the mitochondria, and it's going to be involved in this electron transport system. And when you have oxygen involved, that is called an aerobic reaction. And the fact that it's an aerobic reaction is what's going to allow us to produce all of these ATP molecules. Aerobic reactions can produce much more ATP than anaerobic reactions. And lastly, what else haven't we not accounted for? We haven't accounted for water. And so water is going to be produced during the electron transport system. We're going to have a couple hydrogens that are going to combine with oxygen, and now we're going to make water as a byproduct. Now let's finish off by looking at the details of the electron transport system. And first off, let's orient ourselves. Like I mentioned, the mitochondria has an outer mitochondrial membrane, it has an inner mitochondrial membrane, and then there's this space in between it. And we can see that in this diagram up here. Here's the inner mitochondrial membrane, there's the outer mitochondrial membrane, and then we have the intermembrane space. And again, that's what we're looking at down here in much more specifics. So what's going to happen to start this whole process? These NADH molecules that were carrying electrons are going to drop off those electrons. So we go from an NADH molecule, it drops off its electron, now it goes back to NAD+, and it goes back to those other steps to pick up more electrons. Like it says, my job is done here. Then what happens is these electrons move through these structures in the inner mitochondrial membrane called electron carriers. And these electrons are shuttled through these electron carriers. And as they do, they give off energy. So the movement of these electrons through these electron carriers gives off energy. And what that allows those electron carriers to do is move hydrogen ions from an area of lower concentration up through that inner membrane into the intermembrane space to an area of higher concentration. So we're moving hydrogen against its concentration gradient. And this takes energy. And the energy is being supplied by these electrons being shuttled through these electron carriers. So now what we have is we have a concentration gradient of hydrogen in the intermembrane space. And this represents potential energy. We have a buildup of hydrogen ion that's just waiting to diffuse from an area of higher concentration to an area of lower concentration. Which brings us to this step over here. Now what we're going to do is we're going to involve this structure over here, which is also located in the inner mitochondrial membrane. And this gourd-like looking structure there is called ATP synthase. Now, when you see the suffix ase, what does that mean? That means that it's an enzyme. And what is this enzyme going to do? It's going to synthesize ATP. Now, how does it do that? What's going to happen is there's going to be a channel in this enzyme that's selective for hydrogen. And that's going to allow hydrogen to move from an area of higher concentration down through that channel to an area of lower concentration. And what we're doing by that is we're taking potential energy and we're converting that to kinetic energy. And the kinetic energy of that hydrogen moving through that ATP synthase enzyme gives the ATP synthase enzyme energy. And what's it going to do with that energy? It's going to take ADP, adenosine diphosphate. It's going to add a free phosphate molecule onto it. And it's going to produce, guess what? Adenosine triphosphate. And it's going to produce 32 of those ATP molecules from a single glucose molecule. So this is where all of that ATP is being produced. And just to recap, we're taking electrons that were dropped off by the electron carriers. Those electrons are shuttling through the electron carriers. And as they do, that's allowing these electron carriers to move hydrogen against their concentration gradient from an area of lower to higher concentration. So now we have a high concentration of hydrogen. We have potential energy in the intermembrane space. What's that hydrogen going to do? It's going to pass down through the ATP synthase enzyme, which is going to give that enzyme energy, and it's going to use that energy to convert ADP and a free phosphate to adenosine triphosphate. And that's the whole goal. Now we're making ATP. And just to finish off, let's account for the oxygen. Here we have our oxygen that diffused into the cell, into the mitochondria. Oxygen is going to pick up these electrons here. It's going to combine with a couple hydrogens, and that's going to form our water. So water is being produced as a byproduct. I do think it's worth taking a look at a few more slides just to illustrate the complexity of this process. And we can start with glycolysis. What happens with glycolysis? We take a glucose molecule and ultimately we convert that into 2-pyruvate molecules. But you can see that there's a number of steps that are involved in that process. What else does it involve? What are all these things here? Hexokinase, phosphoglucose isomerase, phosphofructokinase. Well, the suffix gives it away. These are enzymes. And so it requires enzymes to facilitate all of these conversions to get us from glucose to pyruvate. What happens next? That pyruvate moves into the mitochondria, is converted to acetyl-CoA, and then acetyl-CoA enters the citric acid cycle, or the Krebs cycle. And again, you can see this is an extremely complex series of events that are all being facilitated by enzymes. Now, what are the main products here? What are the main products from the citric acid cycle? FADH2 and NADH molecules. These electron carriers that are going to carry electrons to the mitochondrial membrane to be used in the electron transport system. And that brings us to the electron transport system. This is the inner mitochondrial membrane. And these are all the electron transporters. And what this figure is also showing you are potential sites of stimulation of these electron carriers as potential therapy for Parkinson's disease. And here's the research article that you can look up if you're interested in more about that. Here's our ATP synthase enzyme. And this is a much more detailed picture of what this ATP synthase enzyme works like. And if you're interested in learning more about how that actually works, here's a couple YouTube videos that has some video about how that enzyme is actually working. And then lastly, we have focused on glucose as a means to generate ATP, but what this figure is showing is that all of the different macronutrients can be used to enter the cellular respiration cycle and generate ATP. They just do it at different stages of the cycle. So here's our complex carbohydrates like starch, and the digestive system breaks starch down into individual monosaccharides like glucose, and then that glucose travels into the blood, travels to the cells, and it enters the cellular respiration cycle at the glycolysis level. And then over here, we have triglycerides. And what this is showing is that triglycerides can be broken down into the glycerol and fatty acid components, and that each of these can enter cellular respiration. Glycerol enters at the glycolysis level, and then fatty acids move down into the mitochondria and they enter the citric acid cycle. And then lastly, we have proteins, and the digestive system breaks proteins down into individual amino acids. Those amino acids enter the blood, and ideally we want to use those amino acids to build proteins throughout the body, but if we need to, they can be used as energy. It's not ideal, but if the energy reserves are low, we can use amino acids as energy. And what this is showing is that these amino acids can enter at the glycolysis level, or they can move down and enter at the citric acid cycle level. And the whole goal is to produce ATP, convert the chemical energy in these nutrients into ATP, which is a form the cells can use. And here's the structure of ATP, adenosine triphosphate. It's made up of a nitrogenous base called adenine. Then we have a ribose sugar. And then we get the triphosphate. We have the 3 phosphate molecules here. And my question to you is, where is the energy stored? Where's the chemical energy stored in this ATP molecule that the cell is going to use to perform work? And the answer to that is the chemical energy is stored in these chemical bonds, these covalent bonds that are holding the individual phosphate molecules together. And when the cell wants to utilize that energy to perform work, all it does is it breaks one of those covalent bonds, that releases free energy, and that's the energy that the cell is using to perform work. What are we left with? We're left with ADP and a free inorganic phosphate, which are going to be linked back together by the ATP synthase molecule to form ATP again. Full circle.